Good morning and good afternoon. Welcome to session 5 of our Rosalind Encounter Gene Expression Masterclass Series. This is, again, session 5, talking about cell type profiling. And this is Tim Wesselman, the CEO and founder of Rosalind. It is a pleasure to talk to you all. I am introducing Dr. Ryan Friese for this session today. But before I do that, I want to cover a couple key housekeeping rules. And to help guide the session. So this is, of course, GoToWebinar, which means you're all muted. There is a portion over on the side where you're able to ask questions. Please ask questions over there as they come to you during the session. So we have Kuki, who is our Director of Business and Product Strategy, who's on the line. She will be looking at those questions, gathering them up. Where we can provide answers during the session, we will give those to you as we go. Also, I want to point out to you that there are a couple great resources that are provided with the GoToWebinar. If you click, you'll notice there's a couple handouts, which are some downloadable product information. And on those, you'll notice there are QR codes you can scan that'll take you to even more information. So be sure to check out those resources that are provided to you. So now I want to come back to the session today. So these masterclass series are typically reserved for our professional and enterprise customers, but we've decided in the case of this gene expression NanoString focused one to open this up to everyone. It has been really well attended. It has received great response, and I just love the detail and the information that Ryan has gone into. He's got an excellent background and experience in this space. And just to give you a little bit more on Ryan's background. Ryan got his PhD in bioengineering from UCSD. He spent over 10 years with NanoString, working together with customers from pharma to biotech and biopharma and academia. He's an absolute expert in not only the bench side of this, but also in the analysis and in aspects, all things that relate towards NanoString. He's got a deep passion for the science. He's going to talk to you about that today. And I just love the excitement, the passion, and the understanding for what it means to walk in your shoes and also deliver compelling information to you that he provides. I want to balance this with one other very important point. We're talking to you about Encounter, but Rosalind is so much more. So this session is focused on Encounter, but Rosalind is a multi-omics platform. So we do all things with bulk RNA-seq, with single cell, and also in epigenetics, and also proteomics. So there's other things that they'll be covered in upcoming masterclass series. One of them, for example, is a brand new one on geomics. So we have a new professional and advanced capability coming for geomics that is going to really help break the logjam for all of you that are sitting on geomics data, give you a fantastic analysis solution that brings geomics capabilities up to a Rosalind level that allows you to integrate with all the other omics you're working on, not only your encounter data, but also your bulk RNA-seq, your single cell, and again, epigenetics and proteomics data sets. So really looking forward to that upcoming masterclass series, and we're expecting that new geomics capability to release in the next 30 to 45 days. So we're right around the corner finishing off this excellent work on a new geomics experience. Another thing is we're going to have some advancements in our RNA-seq capabilities. So we're expecting radically faster FASTQ to results with exciting new enhancements in some of the intelligence that'll be rolling out. So please pay attention to these updates, and just remember that Rosalind is your trusted research assistant for all things multiomics in the lab today. With that note, I will turn it over to Ryan for another exciting session today, focusing on cell type profiler. Dr. Ryan Friese, the floor is yours.
Thank you, Tim. So appreciate all that warm introduction and expressing a little bit about my background. So this masterclass series, I'm getting a little nostalgic as we're coming to the end here. I've dreamt of doing this while I was actually working at NanoString, but also working on the bench at NanoString at the same time. It just prohibited me from doing it. There just wasn't enough time to focus on developing a full data analysis course. So finally now at Rosalind, I'm focused only on data analysis. So it's coming to fruition. So it's it feels really good for me to come full circle and get this thing completed, which I've had in the back of my mind for years now. So the purpose of me wanting to generate this course and create all this content is because, I really believe that in order to successfully analyze your data and make the correct interpretations and, use that as the basis for your publication or progressing a candidate down your drug development pipeline, you really need to understand how the data are processed, from the very beginning all the way through, pruning, normalization, pathway analysis, and today we're talking about cell type profiling. So I really want to share everything I've gained over the past 10 years or so to enable the whole community just to improve their knowledge of how they're analyzing your data and just pushing all of your important scientific progress forward. The main goal that I'm working on here is just to advance human healthcare and well-being. So that's my motivation, the genesis, why I wanted to do this course. So I know many of you have been here in previous classes, so welcome back. For those of you that are new, there were 4 main objectives that I outlined about what you wanted to learn from this particular class. So I wanted to make sure that you understood that Rosalind is a great resource, Encounter is a great resource, and together you unlock this synergy where you take full advantage of both technologies and they work really well together. And I want you to gain a deep understanding of the analysis principles, like I just said, and the algorithms. Encounter data is beautifully simple. All you need to know is arithmetic to understand how the data are processed. I want to increase your confidence so that you know how things are generated, how scores are calculated, and that therefore you can interpret your data quickly and accurately and correctly. And knowing all this new information, it can open up new insights you might have missed if you didn't have this foundation of knowledge. So if we look across our schedule, we are now June 13th cell profiling, so that's happening today. We have an upcoming session on meta-analysis, which is going to be scheduled for the mid-July timeframe. We were originally going to have meta-analysis for only Encounter, but I wanted to kind of reinvent this and reinvigorate it because, as Tim said, we're much more than Encounter. So meta-analysis has applications beyond only Encounter. You can use it for time course series data, comparing mechanism of action of drugs, integrating public data, multi-omics comparisons, multimodal comparisons. So meta-analysis is a really broad topic. So I want to reinvigorate this session and make sure we can address all of our user base. So that's coming up in the mid-July timeframe. So before I get into cell profiling, let me set the stage a little more for those of you that are joining for the first time to the masterclass. So you're probably familiar with the Encounter technology. From my perspective, having used it and taught it for 10 years or so now, the speed is amazing. You can go from your data, or I'm sorry, your sample to your normalized data, your generated data in about 24 hours. It's very elegant chemistry, elegant instrumentation, and there's a nice minimization of sources of technical variation. Because the assay is so simple, because the instrumentation is so simple, there's very few sources of variation relative to other technologies. That gives you advantages in terms of reproducibility, precision, etc. And I think you probably all know that NanoString is best in class for working with degraded RNA, such as RNA from FFPE tissue. But we're here today to talk about the data. So I love the Encounter data because RCC files, they're very, very small. You can carry them on a thumb drive. Each RCC file is just a few kilobytes, so it's really easy to store, transfer, backup. The data is intuitive. So NanoString is a single molecule counting technology. So when you're looking at the data, you're just looking at single counts of molecules or transcripts. And the last point here is what brings us today is that the algorithms are easy to understand conceptually and mathematically. Taking the time to learn about them kind of on your own is a different matter. But if you take the time and start looking at what's actually happening under the hood, it's pretty easy to understand. You don't need some advanced mathematics or coding capabilities. So I want to kind of demystify the cell profiling for you today. In today's session, we'll be looking in the Rosalind platform as well. In previous sessions, we looked at some of these modules that are available. Last time we looked at that differential expression module and the pathway analysis module. Today, we're going to be looking at the cell profiling module. And we have recently updated our Encounter capabilities. So If you weren't able to join previous sessions, we have a lot of advanced new capabilities, advanced pruning, and custom normalization. I do recommend going back to previous sessions and watching those recordings if you happen to miss those. But today we're talking about SIL profiling. But over all my experience of analyzing data for Encounter, many different applications, software packages, it's my view that Rosalind is the most powerful and collaborative solution for Encounter data analysis that's available. So a lot of these new features I played a role in developing. So kind of my heart and soul is into the Rosalind capabilities for Encounter. So a lot of kind of my influence you'll see in the platform, whether you realize it or not. So before we jump into SIL profiling, I'll leave you with one slide here. And this goes back to that synergy that I mentioned a few minutes ago. So when you pair the Encounter technology, the chemistry, the instrumentation with Rosalind data analysis on the back end, you unlock the full value of both technologies and you get this beautiful synergy. So starting in the lab with your RNA samples, the bench scientists pipettes for 15 minutes to set up their hybridization reaction that runs overnight. That second day, you load your hybridization reaction into your Encounter instrumentation that runs for about 6 hours. And then at that point, you're ready to load the data directly into the Rosalind platform and take advantage of our advanced capabilities and all of our new knowledge capabilities, intelligence capabilities that are not found in any other NSolver or Encounter solutions, I should say. So using Rosalind on the back end of the Encounter technology opens up new insights and it maximizes the value of data, of information that you can extract from your targeted Encounter panel. So with that stage being set, let's jump into SIL profiling. Like I've done in previous sessions, I'm going to pose this question to you to spark your thinking. Even though you may not be directly interacting with me, I want you to think about all the things that I'm saying. So my first prompt to you is, what is SIL profiling? At its fundamental core, why do you want to do SIL profiling? What information do you want to get out of it? In general, what is SIL profiling? So take a few minutes there and think, or a few seconds there, I should say, and think about this, and then I'll show you what I came up with. But just take a few seconds to think about this. So what I came up with is this sentence here. So I try to keep this as high level as possible. SIL profiling at its core, it's a computational method, and we're applying it to bulk gene expression data. In this case, it's Encounter data. And we use it to estimate the abundance of various cell types in an experiment. So that's what it is as its core. But we want to definitely go into SIL profiling specifically for Encounter and talk about how that might differ from perhaps like a flow cytometry experiment. So we want to make sure we are accurately interpreting the SIL profiling results for Encounter. We want to understand how the scores are generated, how the signatures have been developed, so we can make sure we can correctly interpret our data. So let's start at the very beginning. Let's go back to the publication from NanoString. And this was led by Patrick Danaher. He's NanoString's absolutely fantastic biostatistician. He led this effort. And he published all of these results in this publication here on the left. If you haven't read this and you're interested in using the SIL profiling for Encounter, I highly recommend reading it, because as you'll come to see is that understanding how these signatures were developed, the context that they were developed and validated, is really important to understand how they can be interpreted. Signatures, if you're not aware, they can be developed or in many different contexts. So it can be developed so for a certain indication, for a certain sample type in a certain situation. And that context is really important to interpreting the results of a signature or a score. So definitely recommend reading this publication. It'll be well worth your while. It's a really great publication with a lot of nice validation to other technologies. So that being said, let's go into some highlights from this publication to set the stage. So on the right-hand side, I want to show you what the hypothetical result of SIL profiling might look for Encounter data, so you can see the end result before we start talking about how it was actually developed. So this is a hypothetical example of estimating the abundance of macrophages. So our cell type of interest is macrophages. We have some control samples here with this blue box on the left, some condition samples in this orange box on the right. And our conclusion from this figure is that there are more macrophages in these condition samples versus the control samples. So that's the type of information we can get out. But let's go all the way back to the beginning and figure out how we got to this type of score, because the context, again, is really important. So what Patrick Danaher did and his team at NanoString, they went back to the previous literature, of course, and to see what's already been done. So I'm going to just pull out snippets. This is definitely not going to be a complete literature review for you. But the first thing I want to point out here is these excerpts from the publication. So these are directly from Patrick Danaher's paper. So they went back, looked at the current state of affairs. So previously, researchers have done really fabulous work to look at and identify marker genes in immune cells. So they said here, and I quote, previous authors have identified genes specific to purified immune cell populations, and they've used these to quantify immune populations in tumors. So what Patrick Danaher is saying here is that historically, these types of gene signatures, abundance measurements have been calculated based off of signatures from immune cell populations that are purified, that are isolated. So that's kind of the current state or the previous state of affairs before these new scores were developed. And then after the statement, the Danaher team says that, however, these genes were discovered using purified cells and not immune cells taken from the tumor microenvironment. So he really wants to highlight here that there's going to be differences in intratumoral and in vitro gene expression patterns that can compromise these historic signatures in tumor samples. So this is kind of highlighting or alluding to the whole purpose of why NanoString was doing this, and what Patrick Danaher was up to, is that he wanted to make signatures that performed really well in tumors, in vivo, and not signatures that were isolated from purified cell populations. So what I'm showing you here on the right-hand side, this is some of this historic data that the authors were alluding to. So I took this publication number 7 here. It's this paper by Bendea, and what they did in this publication, they used old microarrays and all these different types of purified immune cell populations to try to identify marker genes for these different cell types. And it has been applied historically to tumors, but what Patrick Danaher is saying is that since these marker genes were not identified in the context of actual tumors, how well do they actually perform in real tumor samples? So that's what he's highlighting here. And so I'll have one more slide here on the publication, and that's going to be shown here where they did some validation. So I'm not going to go through all of this. It's some really fascinating work. But to make a long story short, what the team did is that they identified 60 marker genes whose expression levels measure 14 immune cell populations. So the outcome of this work was these nice validated signatures for 14 different immune cell types. And then they did some nice validation to other technologies here. So they said that the cell type scores calculated from these genes are concordant with flow cytometry, with IHC, and they show high reproducibility in RNA from FFPE samples, FFPE tissue, which is key if you're doing clinical research. And it enabled detailed analyses of the anti-tumor immune response in the TCGA dataset. If you're not familiar with TCGA, this is the Cancer Genome Atlas. It's a wealth of information. It's a well-known cancer repository of data, sequencing data. And then this third point here, this additional validation, is that the team then went on to look at immunotherapy data. So these are actual immunotherapy patients, clinical data, and the signatures that the Danaher team developed, they were able to separate responders and non-responders according to their therapy, and provided this intricate picture of the effects of checkpoint inhibition. And this last point here I want to bring up is that the team, again, kind of cautioned that previously reported genes to be enriched in single cell types have co-expression patterns inconsistent with cell type specificity. So this is another caution in using these old historic cell types that might have been, old historic signatures or scores that were developed from purified cell types in isolation in vitro. They may not perform well directly in tissues in your samples of interest. So those are some highlights I wanted to bring out from this publication. Again, it's a fascinating read. This is only a snippet. I strongly recommend reading the whole thing if you're interested in applying these types of results to your data. But what I'm showing on the right here, I pulled out two figures that the Danaher team generated. This, I know it's very small. The details don't matter here. But this is a figure showing the validation of the B cell signature across the dozens of different tumor types within the TCGA dataset. So in the TCGA dataset, there are many different indications of cancer. And so the Danaher team, they were validating their B cell signature across all these different indications to ensure they had a robust signature. Down below, this is the comparison or the validation of Danaher signatures versus IHC, and then Danaher signatures versus flow cytometry. So to make a long story short, there was some really excellent work that was put into this to identify what was the current state of affairs, identify how they can be improved or how it can be improved to make sure that the scores are validated to perform well within tumor samples. So that's why I'm saying that the context is really important with how these signatures are developed. If you're using the immune cell scores from NanoString, they have been validated and developed, or I should say developed and then validated actually within tumors. So if you're interested in immunotherapy and profiling the abundance of cell types in your tumors, these signatures have been validated for that purpose. So that's really important to know. Final thing I'll say about the publication is this conclusion here in the bottom left, the final words from the authors. Due to their concise gene set, computational simplicity, and utility in tumor samples, these cell type gene signatures may be useful in future discovery research and clinical trials to understand how tumors and therapeutic intervention shape the immune response. So this goes to the high level purpose of why the team wanted to develop these signatures. They wanted to empower researchers to, in their discovery, but also in their clinical trials to improve therapeutics and our understanding of cancer and tumor biology. So that's the publication, that's the foundation for all of these signatures, all of the cell type profiling. Now let's go into how are they interpreted and how they're actually calculated. So again, here's another example of a hypothetical score. This is a macrophage cell type abundance score. We can see the macrophages are more abundant in our condition samples versus our control. Let's make sure we are interpreting these scores correctly. So I put together a table in the next slide, providing some guidance on how to interpret these scores. And we'll see some scores in a few slides here. But on the left, I wanted to bring your attention to these two situations to make sure you are not incorrectly interpreting the cell type signature scores or the cell type signatures. The first incorrect interpretation is that these scores are actually calculating the number of cells in a given sample for a specific cell type. So you're not looking at sample number 1 and figuring out how many or what is the number of T cells in that sample. That is not what's happening here. It's because all of these gene signatures are based off normalized expression of genes. We don't know the relationship between gene expression and the number of cells and the number of transcripts. So we're not quantifying number of cells. Also, what we're not doing is we're not enabling the comparison within the same sample between two different types of cells. So we can't look at sample number 1 and say, what is the ratio between CD4 cells and CD8 cells? That's not a correct interpretation of these signature scores. You cannot compare 2 or more cell types within the same sample. The correct interpretation is shown on the right-hand side, and this shows you that what we are doing is comparing relative abundance of a cell type, a specific cell type, across different samples. So you can look at your T cell scores and look at that T cell score across all the different samples in your data set and see relatively to each other, does the abundance of T cells change from sample to sample? Does it increase? Does it decrease? Is it more abundant in controls or treatment, etc.? So the scores give you a relative abundance of any given cell type across samples. The next kind of tip here in terms of interpretation I showed here on the right-hand side, and that is these scores, they're in the log2 scale. So what that means is that when you're looking at a difference in scores, let's say between 2 samples, so I'm going to make this just very simple hypothetically. So in sample number 1, your T cell score has a value of 1, just hypothetically. And sample number 2, your T cell score has a value of 2. Now, since that is in it's log2, it means there's a 2-fold increase in abundance. Let me make another example here. So let's say your score is, in sample number 1, it's 8. In sample number 2, your score is 9. So the difference there is actually a unit value of 1, but since it's a log2 scale, it represents a doubling. So just keep that in mind, and I'll talk about this more a little later, is that these scores, they're all in log2 scale. So whenever you see a difference value of 1, it represents a doubling in the abundance of cell types. So that being said, let's talk about where you can find the cell type scores and the profiling and the signatures according to NanoString's catalogs of panels and what cell types are available. So I briefly put together this table. This is not comprehensive by any means. So NanoString is going to have the most up-to-date listing of their panels, of course. But as an example here, there are some panels off the shelf from NanoString, such as pan-cancer immune profiling, immuno-oncology 360, breast cancer 360, where you can profile the abundance of 14 different immune cells. As a complement to that, they've focused also on neuroscience. So they have a neuropathology panel where you can profile the abundance of neural cells. They have a neuroinflammation panel where you can profile the abundance of neural cells and immune cells. And so they have, additional panels as well that have these immune and neural capabilities. But just want to bring your awareness that there's also this neural type of cell profiling, which I didn't talk about in the Danaher paper. That came a little later. But there's this nice complement of immune cell profiling and also neural cell profiling in the NanoString catalog. When we think about the cell types that we can profile, on the immune side, we can profile 14 different cell types. So these cell types are shown in a column on the left. The representative marker genes are shown here on the right-hand side. So this is your selection of immune cell types that can be profiled. If you're focused on neurology, neuroscience, you have access to profile these different cell types: neurons, microglia, astrocytes, oligodendrocytes, and endothelial cells, and you can see their respective marker genes. So let's talk about how you go from one of these cell types and then its signature genes and use that to create a score for every sample in your data set. So to do that, I went to the NanoString Advanced Analysis Manual, extracted some excerpts, and then try to translate that into some basic math and some plain English so we can understand what's happening. At its very core, we are simply looking at normalized expression values. So this is a quote directly from the NanoString manual. A cell type's abundance can be measured as the average log scale expression of its characteristic genes. So to translate that a little more, so it's a little more clear, let's look at this example below. Let's say we want to profile the abundance of macrophages in my sample number 1. NanoString has validated a signature of 4 genes to do that, so these 4 genes. So let's look within sample number 1. We can look at these 4 genes. We can look at its normalized expression. So this is the normalized expression in linear space, linear counts. But as I mentioned earlier, all the analysis is done in this log2 space. And so we basically transform the data to log2 space here. We just take the log2 of 512, and that's 9 in this example. So we convert the data to log2 space. And then to calculate the abundance score of macrophages in sample number 1, we take the arithmetic mean of all signature genes. So for these 4 genes, CD163, CD68, CD84, MS4A4A, I look at the log2 normalized counts. I simply take the arithmetic mean of all 4 of these genes. This is the formula here. So just the sum divided by 4, and that equals 8.99. And that's it. So for each sample, each cell type of interest, we look at the signature genes, we take the log2 normalized counts, and then we take the arithmetic mean, and that equals your score. So that's pretty simple to understand. It's this average expression of these cell type markers. But it's a little more complicated than that. So let me go to the next level of analysis here. Let's talk about the assumptions behind these genes and whether or not all of them are used all the time in all situations. So further down in the nCounter Advanced Analysis manual, you'll find some additional excerpts describing this methodology. So they have a section called Automatic Screening of Failed Cell Type Specific Genes. And there's this quote here that I want to read you. It says that the algorithm used to identify appropriate marker genes and exclude badly behaving cell type specific genes from estimates of cell type abundance is detailed below. So I underlined this section, this sentence, or these words here, exclude badly behaving cell type specific genes. And there's part of the algorithm is to look at all of the genes in a signature for a cell type. And if any of those genes are not performing well in your specific data set, then we actually kick that gene out of the signature. So I'll show you what that looks like in the next slide or two here. But I want to kind of prime you at this point to know that we don't always include all of the genes for a cell type. So I'm going to go back and let's look at our macrophage signature. So if we apply our algorithm and we assess that this last gene is not performing as expected, then we're actually going to kick it out of the signature. And when we estimate the abundance of macrophages, we're only going to look at these 3 genes, and we're going to exclude this 4th one. So I'll show you how that's done in a couple minutes here, but I just want to let you know that that's happening in the back end, even if you're not familiar with that, or if you haven't heard of it before. So to do this, what NanoString does is they define a similarity metric between 2 candidate cell type specific genes. And here are the important assumptions. So for every single gene in a signature, there's these assumptions that any 2 genes in a signature, they are both specific to the same cell type. So that makes sense. If we're looking at macrophages, any 2 genes, any pair of genes in that signature should be specific to macrophages. So I think that's an important assumption. And then the second assumption here is that both genes are consistently expressed within the cell type. So within, let's say, macrophages, we want, as an example, both CD163 and CD68 to be specific to that cell type, specific to macrophages, and also consistently expressed. So these are the assumptions, whether you realize it or not. These are the assumptions underneath all of the genes in the signature and how they perform when we are actually calculating the scores. As an example of this, let me pose a hypothetical and get you thinking about these assumptions and how they might be violated. So we could have this situation where we had a tumor sample. We have T cells in your tumor, and your T cells are expressing, let's say, gene A. So in this hypothetical scenario, the number of T cells in your tumor can double, and that will result in the gene expression of gene A to also double. So that's the scenario where both of these are going to be, these assumptions are held true. There's another situation where, for example, you have your T cells in your tumor, and the number of T cells does not increase. So the number of T cells stays the same, but the expression of gene A actually doubles. And this is because there's some sort of transcriptional activation of gene A specifically that's causing its expression to increase kind of independently from the number of T cells that are in the sample. So in that situation, this assumption would be violated here. So it's kind of like a deep nuance to these assumptions, but it's an important one because the way that NanoString calculates these and looks at the similarity of genes, we want to know that both of these assumptions are true. So this will make a little more sense when we start looking at the next screen here. Now the final thing is that if we look at any 2 genes in a cell type, we want them to be highly correlated with a slope of 1. So this makes sense too. So, if these assumptions are true, if we look at any 2 genes within macrophages, they should both be specific to the cell type. They should both be consistently expressed within the cell type. And then they should have a high correlation with each other with a slope of 1. So these are kind of all the underlying assumptions going into the genes within your signature. So let's look at this in a little more detail. I'm not going to go into the math in too much depth. There's some statistics. I'm just going to show you the equations. I'm not going to show you how they're calculated. But essentially what's happening here is that for any 2 genes, let's say in a macrophage signature, what the algorithm is doing is actually calculating a statistic similar to a Pearson correlation. So we basically want to see, like, pairwise of every gene in a signature, what is the correlation of all of these genes to each other, and is the slope close to 1? And if that's true, all of those genes are going to be included in the signature because they meet all of the assumptions. So This may look a little complicated. I'll show you an example of this in the next slide here. But for every pair of genes in a signature, we calculate this similarity, and just for the purposes of this, just think of this as a Pearson correlation, also assessing how close the slope of the correlation is to 1. And these are direct quotes from the NanoString manual saying that the similarity function, it equals 1 when 2 genes are perfectly correlated with a slope of 1, and decreases for gene pairs with low correlation or slope diverging from 1. And then down below, since many biologically related genes will exhibit correlation unrelated to a shared cell type, it is important to apply a more stringent measure of similarity than mere correlation. So all of this is saying is that within a given gene signature, we look at all pairwise combination of the genes in there. We assess essentially what is their correlation, and then what is the slope. We want all pairwise combinations to have a high correlation and a slope close to 1. And when that's true, all of the genes of the signature are performing robustly, and all of them will be used in the score calculation. To give you some visualizations of this, we have these images here on the right-hand side. So let's pretend we're focused on our macrophage function score again. We have gene 1 on the x-axis, gene 2 in the score on the y-axis. And so what we're seeing here is that these 2 genes have a really strong correlation to each other, and the slope is 1. So these 2 genes are performing robustly to assess macrophages in your data set. Each dot here is a sample if it's not clear. So it's the expression, each dot is a sample, and it's showing you the expression of gene 1 versus gene 2. So there's this other example here in the middle where you're looking at, let's say, a different gene 1 and a different gene. Now this looks like it's gene 3. These are both genes in the macrophage score. In this case, they're not correlated at all. And there's no slope close to 1 at all because there's no correlation. So in this case, one of the genes is not performing well and should probably be kicked out of the signature and not used to estimate the score. Another example is shown here on the right-hand side, where you have gene 1 in the macrophage signature versus gene 4 in the macrophage signature. You see there's this nice correlation, but the slope is not 1. So one of our assumptions is that we want the slope of the correlation to be as close to 1 as possible. This is not meeting that assumption, so this gene 4 is going to be kicked out of the signature. So let me show you a table on the next screen to make this a little more sensible here. So we have our macrophage our macrophage cell type signature on the left. In this table, we have 4 genes. For each combination of genes, so CD163 versus 163, of course, it's 1. The similarity is 1 because it's, the identity. But then we want to look at 163 versus CD68, CD163 versus CD84, CD163 versus MS4A4A. And so we look at all possible combinations of this similarity. So again, think of similarity as looking at the Pearson correlation, but also wanting the slope to be close to 1. So what we can see here is that CD163 and CD68, 0.94, this is really high. We want both of these genes to be included in the macrophage estimation. CD163 and CD84, 0.88, this is high. We want these genes included in the macrophage estimation. Same with CD68 and CD84. But when we look at MS4A4, these values are actually really low. These gene pairs are either not correlated or the slope is not close to 1. So for that reason, MS4A4 is going to be kicked out of the estimation of macrophages. And when we estimate the score, there's only going to be these 3 genes that are going to be used. So I know that was a little complicated, and I didn't detail the math very much. But just to kind of summarize what's happening is that there's this QC process in the background. Within each cell type, we look at all of the pairwise combinations of the genes for that cell type. We assess how strong is the correlation, how close is the slope to 1, and if any genes are not performing well, in this example, MS4A4, that specific gene is kicked out of the cell type signature and it's not used in the abundance estimation. So I'll show you what this looks like within the Rosalind platform here in just a couple minutes. I think I have one more slide and then we'll go into Rosalind so we can see some real world examples and some real data sets. The last thing I want to point out, and I'm going to show you in the next slide, this long equation. I'm not going to describe it very much at all, but we are actually calculating a p-value that's showing you the performance of the similarity estimate. So for each cell type that we generate, you'll get this QC p-value, and the p-value is telling you is within the gene, within the cell type, I should say, within the cell type, how similar are the genes to each other? Do they have strong correlations? And then are the slopes close to 1? So that's going to be shown here in this next slide. So at the top, this is an excerpt directly from NanoString's description of this in their manual. So it can be a little complicated, so I'm not going to talk about it. But a long story short is that they run a permutation statistic to evaluate how robust these different cell type signatures are performing. And again, by robust, I mean, are all the genes in a cell type, do they have strong correlation? Is the slope close to 1? So this is an example of what it might look like within the Rosalind platform. For each cell type, for example, T cells here, we run this QC p-value, and the lower the p-value, the more robust the signature is performing in your data set. So there's probably some questions about this. I'm going to show you what this looks like within the Rosalind platform, show you how to interpret everything, and then I'll pause for some questions. So let me transition out of PowerPoint. And we'll go to a data set. So the first data set I want to show you is a breast cancer data set. So what we're looking at here is going to be different molecular subtypes of breast cancer. If you're not familiar with breast cancer, there are different molecular subtypes that can be used to define the biology of the tumor itself. So if you're not familiar with these, NanoString commercialized them as the PAM50 signature score, but the different molecular subtypes are luminal A, luminal B, HER2 enriched, and basal-like. So in this particular data set, we're looking at some different molecular subtypes of breast cancer, and we are assessing them with the immune profiling panel. So when we look at the samples or the experimental design, we see that I think there's about 40 or so samples in here, but we'll see that, some of these are normal control samples. So we have some normal control samples, and then some of the samples are luminal B subtype, some of them are luminal A subtype. So in this particular data set, we are comparing samples of normal controls versus luminal A molecular subtype for breast cancer versus luminal B molecular subtype for breast cancer. So it's normal versus different types of breast cancer, essentially. When we go to our results for cell profiling, we are brought to a page like this, and you'll see on the left-hand side the list of cell types that are profiled in the panel. So again, this was the immune profiling panel, so we're able to profile the abundance of 14 different immune cell types. You see those cell types listed here in this column. To the right of that is that QC p-value I just mentioned. Again, this is looking at within each cell type, so let's say within T cells, how correlated are the genes in this cell type score to each other? How strong is the correlation, and how close is the slope to 1? So these p-values are low. So that indicates in this particular data set, the performance of the T cells score is going to be robust. We'll see at the bottom here, there's some of these signature scores that have, P's of 1, indicating that perhaps in this data set, these cell types are not performing robustly. So you definitely want to proceed with caution according to your different p-values here as well. So we can do some interrogation of specific cell types, but what we're looking at now is a heat map of all of these cell types selected together. So we are clustering based off of the relative abundance of all these different cell types across all my different samples. If you used Rosalind before, you know that you have something called an attribute. So in this data set, I have different subtypes of breast cancer, so I can stratify my heat map by different subtypes here. So I can do that by coloring the subtypes. So you can see that these goldish colors are luminal B samples, the orange samples are luminal A, and then these blue samples are my normal control. So if we take a step back and look at this heat map, clearly on the right-hand side, there's this subset of samples that have this really strong kind of immune abundance, immune infiltration, which is represented by the red color. And on the left-hand side, there's also some tumor samples along with my controls that have this low abundance of immune infiltrate. So that's a really interesting conclusion just by itself. We have some samples, high immune infiltrates. It looks like in the middle, there's this cluster of samples that have this kind of medium value of infiltrates. And on the far left-hand side, there's samples with low. that includes both normal controls, but also some luminal A and luminal B. So maybe that's interesting. Maybe that can be used to predict responders versus non-responders to an immunotherapy. If I'm interested in certain cell types, I can certainly click on them. So if I want to look at, let's say, only T cells, I can click on that. And now what we're looking at is this box plot, and it's showing me the abundance of T cells in my different samples. So I have my normal controls here, my luminal A's here, and my luminal B here. Each circle, each dot is a sample. The y-axis here is my abundance score. Again, this is my log2 scale. So a change of 1 on my y-axis represents a doubling or a change of twofold in abundance of the cell type. So my conclusion looking at that is that, luminal A, it looks like it has higher T cell abundance versus my normal controls. That makes sense. And, luminal B versus A, perhaps those are equivalent or very modestly different. So I can definitely go through each one of these one at a time, look at all my different cell types, how they vary in abundance across my different biological groups. So in this case, dendritic cells, my antigen-presenting cells, there happens to be fewer in my luminal A and luminal B in my cancer versus controls. So maybe that's interesting by itself as well. That's a really interesting signature or biomarker. Maybe that's gonna be useful to help me in my immunotherapy development. So there's a lot of interesting things and kind of next steps you can take based off of this data. In addition to looking at this box plot, which is really the summary view. So we're looking at within my normal controls here, what is the summary of the abundance of T cells in normals, or in luminal A, when we look at the box plot, what is this summary of T cell abundance in luminal A? I might be interested in looking at specific patients. So we know, there's a lot of natural heterogeneity in immune response within and across specific patients. So instead of looking at this summary view with box plots, I can change this and toggle it to actually look at patient level view. So this is really interesting because let me go back to my T cells here. So all these blue ones are my normal controls. All of these goldish ones on my end are my luminal B. And in general, there appear to be fewer T cells in controls compared to my luminal B. But there's a lot of heterogeneity in human patients, human samples. So this one sample, whatever it may be, has low T cells, relative to the other luminal Bs. So maybe this patient is a responder. Maybe this patient is destined to be a non-responder to my immunotherapy. I'm not sure, but this is a really interesting result. So I definitely encourage you to look at each cell type one by one, and then look at the box plots to look at the summary view, but then also definitely look at the bar graphs, because you can find really interesting patients and look at the heterogeneity across your different patients or your different samples. Okay, so last thing I'll show you is we have some data we can look at. So right now I've just been showing you visualizations. Of course, we can pull out the actual values themselves. So I'm going to go back to this toggle here in the top left and toggle to this table. Right now I'm looking at only T cells. So you can see on the left-hand side, I've selected T cells. That's why I see only T cells here. I'm going to click select all, and then I'm going to generate a table including all cell types. And what you'll see is that you have in each row the name of your cell type. You have the maximum difference in score across your samples. What is the highest score across all your samples? What is the lowest? You have that QCP value indicating in your particular data set, how robust is the T cell signature performing? Again, I'm just going to be a broken record here, but this QCP value is assessing within the T cell signature in your data set, how correlated are the genes and how close is the slope to 1. So you'll see that p-value for all of your different cell types here. And then what you'll also see is the inclusion and exclusion of specific genes. So if you remember when I was showing you the PowerPoint slides, I showed you that if any gene in a signature does not have high similarity or high correlation to the other genes in the signature, then it's actually excluded from the signature because it's not performing robustly in your data set. So in this particular data set, when we're profiling T cells, we are using these genes to profile the abundance of T cells, but we are excluding these ones here because in your data set, they are not performing robustly. And so you can get that information across all of the cell types that we have access to here. The other thing I'll mention in terms of the exclusion is that, and you'll know this if you attended our earlier master classes, is that we apply some pruning to low expressed genes. So I'm going to make a hypothetical here. Tregs. So, if FOXP3 is the marker gene for Tregs, but this gene is actually not expressed in your data set, and the gene is pruned, if it's removed, FOXP3 is removed, then you don't have the ability to profile Tregs, of course. So if this gene is not expressed, then it can't be used in the profiling. That just makes sense. And so if you've used our advanced workflows and you get to the custom pruning section, you may see some of these genes in these signatures being included or excluded based off of their expression. So when you look at these excluded genes, they could be pruned for low expression, or they could just be excluded from the signature because they're not highly correlated to the other genes in the signature. Okay, we have about 9 minutes left. I'm going to show you one other data set, and then I'll stop for some questions. What I just showed you was a data set of tumors. So these are breast cancer tumors and some normal tissue controls. So we are looking at essentially immune cells within the context of the tumor. So we have this, in this tissue, we have a mixture of many, many different cell types. And so we would expect in that case to see many of these cell types within my data set. We would probably expect T cells in my tumor. We would probably expect macrophages. We would probably expect B cells. So it makes sense for us to have an expectation of seeing many of these different cell types expressed and performing in my data set. But this goes back to what I said earlier in the very beginning of my presentation, is that we want to understand the context of the signatures and also the experiment itself. So since these signatures were developed in the context of the tumor microenvironments, they were validated with the TCGA data set, it makes a lot of sense for these to be performing really well in my tumor samples here, in my tissues. I have another data set I want to show you, and that is a cell culture. So while I'm transitioning over here, and I'll mention this is a monocyte cell culture. So when we're looking at the cell type profiling results for my monocyte cell culture, what do you expect? So think about this for a few seconds. What do you expect in terms of the number of cell types that we're going to be able to detect and how robust they are in terms of their QCP value? So let me add an additional caveat here, is that this is actually a stimulation. So in this experiment, we have this time course. So we start with macrophages. I'm sorry, I think I said this wrong. We start with monocytes. So we start with monocytes and then we stimulate them with PMA to differentiate to macrophages over time. So in the very first sample we have these pure cell culture of monocytes, and then we differentiate them to macrophages. So how is cell profiling signatures and scoring going to work in that context? So let me go to the results and show you. Hopefully you've had enough time to think about or anticipate what we're going to see. First thing on the left-hand side is that you'll see there's just fewer cell types available. And that's just because we're essentially only looking at 2 cell types in this data set. We're looking at monocytes, we're looking at macrophages. There's some transition in between the two, but we wouldn't expect most of these cell types to be profiled because it just wouldn't make sense. They're not present in my experiment. The other thing you'll notice is that none of these QCP values are going to be less than 0.05, are going to be quote unquote significant. And that kind of makes sense too, because if we look at this experiment, we have different time points. So we have 0 hours, 12 hours, we have 6 different time points here. So at the very beginning of the time point, we have monocytes. At the very end, we have macrophages. So across this entire data set, none of these signatures are really going to perform robustly because the cell type is essentially changing. So just think about the context of your experiment when you are applying these cell type scores. When I'm looking at this data set, since it's basically only 2 cell types, cell culture, what do I expect in terms of the cell profiling results? I probably only want to focus on my macrophages here. Even though the p-value isn't quote unquote significant, I think this data is still interesting and useful. Just a reminder, this 0.382, this looks across the entire data set, how robust the signature is performing. Obviously, it's not going to be robust across the whole data set because macrophages are only present in the end of the differentiation. But when we look at the data here, the scores, this is a really fascinating data set to me, because to me, what I see is differentiation from monocytes to macrophages. So in the very beginning of my experiment, I have a monocyte cell culture. So the score for macrophages should be very low. And that's what we're seeing here. And then as we stimulate with PMA over 96 hours, we start to see this shift and increase in abundance as macrophages start differentiating into, as monocytes start differentiating into macrophages. So for me, even though this p-value is not significant, I think this data here is really, really interesting and really fascinating, and is actually robust. So the final thing I'll leave you with is always look at these p-values, but think about them in the context of your experiment. If you're just looking at tissues or tumor samples with some treatment, that's one situation. The context here we're looking at is a cell culture, isolated cell populations where you might not have all the cell types expressed, and maybe the cell types themselves are actually changing. So always take these with a grain of salt and make sure you understand how these p-values are calculated and what the scores mean. So I think we have 5 minutes left, so I'll pause there for some questions. Thank you everyone for listening.
Yeah, thank you, Ryan. So this is the Q&A portion. So if you have a question, please make sure you go to the question section on the right and submit your question there. We already have some submitted, and so we'll go through those here in just a quick moment. Ryan, I thought one other thing you may want to comment on, and this goes back to some of the history of the Rosalind solution for nCounter. Cell type profiling was added early on with the starter subscriptions, but we just recently added several enhancements that were the top requests from your old team. Would you mind taking just a quick moment and summarizing what has changed now that we have the professional version in cell type profiling?
Sure. Yeah, so the most requested features that we've added is visibility to this QCP value. So historically in our nCounter starter, this was not available. Now this is front and center directly next to the cell type name. The other piece of information that was requested was going to be, when we look at the table, the included versus excluded genes, and also the p-value. So all this information is now shown directly in the interface on the page, and you can also download it as well. So those are the new enhancements. Also, we are now showing all cell types, regardless of p-value. So historically, when we had developed this in conjunction with NanoString, there was a request to exclude cell types if their p-value was greater than 0.05. But we thought about this a little more, and we recognized the value of including cell types even if the p-value is greater than 0.05. So now when you come to results, you'll see all cell types, all p-values, and all genes included and all genes excluded from the scores.
Thanks, Ryan. That will feed well into this next question, which was, what's the difference between cell type profiling versus other biomarker profiling? For example, microRNA using nCounter, specifically Rosalind, or the latest panel for microRNA profiling versus looks like NanoString and Solver or Rosalind.
Trying to think of the context here with microRNA, but I think in general, cell type profiling is using, so first of all, it's designed for nCounter gene expression data. So that's where it was developed and validated. The importance here is that these are validated signatures. I tried to emphasize that when I was talking about the Danaher publication that first presented these, but these have been extensively validated in the TCGA data sets. They've been compared to flow, to IHC. So these are actually validated biomarkers, and they are more robust than simply looking, perhaps within various databases or previous publications where some marker genes have been identified as being associated with cell types, but they haven't been validated. So the validation aspect to these cell types and these scores is really important. I don't want to underestimate that. It's really, really valuable, the work that the NanoString bioinformatics team put into these. It's not simply finding marker genes. There was a lot of validation to make sure these perform robustly. So I'm not sure if I answered that question in regards to microRNA, but I think that's a good place to end.
Yeah, it is. And thinking about your answer, it occurs to me that there may be one other area that'd be useful to touch into. So actually 2 areas. One is a follow-up question for you, and I'll try to address the other one. And it's this point that obviously there is a very robust solution here in terms of how NanoString has characterized these signatures. But also Rosalind provides an ability for you to develop your own lists, which in effect can be a form of signature. So you'll see that tab right down at the bottom, there's a list tab. And what's interesting is then you can select and create your own collection of these genes and see their performance within your different comparisons and look at those results. And so this usage of gene lists allows you to effectively create your own interesting view and dynamically see these as you're looking through your comparisons, exactly as Ryan's describing here. Now, this gets really interesting because you have consistency. So as Ryan's saving this list here now, when you go across your data sets, and not only for you individually as a scientist, but also for your team, you can share these lists. So then you can easily select it from this menu, just as Ryan has done, and see those results in every data set that you're looking at, every comparison you're looking at. And then one other important delineation. For our enterprise subscriptions, many institutions have developed your own signatures. We bring those into Rosalind, and just where you see NanoString annotations there on the right, we will calculate and display your own signatures there. And so you can look at those. We give you a GSEA score and a p-value and p-adjust for your signature. So we help you really take the knowledge you've been already developing internally and bring that together with Rosalind Intelligence to give you some of those answers. So I think that last question was trying to contrast some of cell type profiling versus other biomarker profiling. And this range of capabilities is related there. There is another very important feature that has been requested that we'll be adding to our biomarker profiling or biomarker identification modules within Rosalind, and it's the single sample pathway scoring. Did I say that right, Ryan?
Mm-hmm.
So that is one other variable that may be important here, and I'll save that for you, Ryan, to say more if you want to. But the next part of this that's probably worth touching on is just contrasting NSolver's ability to do cell type profiling versus Rosalind's.
Sure. Yeah, let me go back. And I'm going to go back to my different data set. It's a little more comprehensive. So yeah, the beauty of the interactive cloud solution kind of comes to life when you're looking at cell profiling results. So it highlights a lot of really nice things about doing this type of analysis in Rosalind. So we have full interactivity of the results, and you can essentially make figures on the fly. So you can, look at a certain cell type, of course. You can quickly cycle between all of them. You can change between box plots, bar graphs. You can get this hover functionality. You look at the data. You can, of course, look at table views as well. And then if you weren't aware, you can actually make box plots on the fly. I'm sorry, heat maps on the fly. So let's say in my breast cancer samples here, I'm really interested in T cells and perhaps CD8 cells and cytotoxic cells. So I'm just selecting these one at a time. Maybe I can even throw dendritic cells in there, and I can see how my different samples relate to my different biomarkers here. So I have this kind of novel biomarker I just created. It's my breast cancer data set. Let me do this one more time to show you. I selected T cells, cytotoxic cells, CD8 cells, this new biomarker on the fly, and there's this huge division where on the right-hand side, there's this set of samples that have, it's bright red. So there's this high infiltrate T cells, cytotoxic cells, CD8 cells, and on the left-hand side, clearly these samples are blue, and they have this low infiltrate of these cell types. So I just created my biomarker on the fly to segregate my samples versus high versus low TILs just in a matter of seconds. And so you can do this type of interactivity, this nice quick biomarker discovery in the cloud-based platform that you can't do in the static NSolver view.
Ryan, before you leave the screen, one more quick one. Rosalind has this capability to also do it in an on-the-fly screening by selecting the reference. And I'm just wondering if that would be relevant in the context of cell type profiling, where, let's say it was based on the sample ID, and you knew you wanted to use one of your controls. So let's take by sample. And I'm thinking if you use one of those ones towards the left side as your reference, I can't see exactly on my little screen some of those sample names, but there you go. But if you selected one of those, then of course we immediately adjust and set that to 0 and give the relative change in relation to those. Do you think this instant screening type of capability is useful for cell type profiling?
I think it is, yeah. Because the foundation of the score is that it's relative. It's a relative abundance. So we're not looking at absolute numbers of cells or anything like that. So the score by nature is relative. So being able to change the relative baseline of your visualization is quite powerful, just like you said, Tim. So in this example, let me 12962. I'm not going to be able to find that quick enough here, but I essentially set this one sample to my baseline reference, and then everything is in relative abundance relative to that one sample, or I could do by subtype. So I know I have some normal controls in here, so let me color everything relative to my normal controls, and we can see how that actually impacts the visualization as well. So I think this reference comparison is quite valuable for the cell type scores because by their very nature, they're relative to each other.
Awesome. Well, I think we thoroughly addressed that one. Let me take you to the next question. Thank you for that. Thank you for the question, by the way. So the next one is number 1. Do the dots on the plot tally with the number of samples in the comparison groups? And number 2, since the cell type profiler doesn't compare between cell types within the same sample, can we tell which cells are most abundant in our samples?
On the first question, each dot here is one sample in a sample group. So here's my normal control samples, and so each black dot is one sample. So the tally is really shown by the box plot itself. So if you know the box plot visualization, the center line here, horizontal line, is the median. We have the 25th and 75th percent quartile, and then the range here shown by the whiskers. So the tally is represented by the box, whereas each dot here is an individual sample within this entire group. And then for the second part of the question, there is no way to really know which cell type is the most abundant, at least not in a very quantitative way, because if we think about this, let's go back and look at the genes. The cell scores are really not meant to do this just by the nature of the technology. Give me 1 second. So what we're looking at here is these different cell types represented by all these different genes. Some of these genes are high expressors, some are medium expressors, some are low expressors. So there's really no way to relate the expression level of the genes to an actual, relative abundance. It's very difficult. In T cells, CH2D1A may be very, very lowly expressed, whereas in NK cells, XCL2 might be a very, very high expressor. So there's no way to do kind of that relative thing. I think that answers the question.
And at risk of stating the obvious, this would be a great place where the multi-omic aspect of Rosalind could be helpful, because if they were to then run a single cell analysis and then utilizing Rosalind's automatic cell type identification on single cell, we would help them look for those cell types and be able to see an abundance of cells expressing within those cell types from the single cell analysis.
Yep, yeah, that's a really great point. Yeah, very true. Yeah, if you want to do the multi-omics integration, that would be a really great way to do it.
So let's see, the next question is, I am curious, is it possible to access codes about how the cell type profiler works, understanding it is run as an R-based program?
So we're not able to share the code itself. All of the calculations are actually available, kind of a description of the math of the calculations are available. That's all open, but the code itself is not. We get that code directly in our partnership with NanoString, so we're not able to share the code.
Okay, and let's see. I don't see any other questions here, so I think at that point, when we are about 10 minutes over, we're probably ready to wrap up. So Ryan, I'll just say on my part, thank you for another amazing and engaging session, and I'll leave it to you to do the rest of the close.
Thank you all for joining. This wraps up the Encounter specific portion of our masterclass. So I think if you joined all 5 sessions, you probably have 5 hours of training under your belt. If you think about many researchers that use the Encounter platform, they might get 45 minutes or an hour total with someone. So just kudos to all of you for joining and investing in yourselves and your education. I think this is going to pay great dividends for yourself in your career, in your all the programs, all the drug programs you're developing, your publications. So our next session, again, in the mid-July timeframe, is meta-analysis. In my opinion, this is probably one of the most powerful features of Rosalind that's the least understood because it has so many great things it can do. So I want to talk about that in the next session. It has applications for multiomics, multimodal comparisons, time course data. You can even use it in QC, which I alluded to in one of the previous master classes. So I would definitely hope to see all of you in the next session on meta-analysis because that's going to be the final piece that brings everything together for your data and your results. Thank you all again. I will see you in the next session.
Hey Ryan, really quick, before you end the session, you mentioned the amount of time and training that's provided here. It's probably worth also mentioning for folks that are interested that you are planning on turning this into a form of certification that helps them share with others that they have developed a deeper expertise here and they have gone through these programs. So it's something that it is an investment into yourself and your abilities, and we want to help you share that with others. Do you want to say anything more on that before we close?
Yeah, just to reiterate that, yeah, I really want to turn this into a formal course. This has been an investment in time by all of you. And so to thank you for joining, now I want to turn this into official certification that you can post on LinkedIn, on your resumes, and kind of, elevate yourself among those who haven't had this training, because I think it is going to give you an advantage in everything you do. So look out for that soon, and I'll let you know when that's available. But thank you all.
Thanks, Ryan. Have a great day.